Hello everyone, I hope you're doing well. I'm Bhagyashree and we are learning about the cell structure and function. So you can watch the previous videos for a better conceptual understanding and with the clarity in your mind so that you will be able to know in more detail about cell structure and function. And if you like the video, don't forget to click on thumbs up button below. Also click on subscribe button for more video updates. And if you have any doubts, feel free to ask in the comment section below. Also, if you want me to cover any specific course or topic, you can suggest me in the comment section below. So in our previous video, we learned about the sterols, phospholipids and glycolipids, lipid bilayer. So this is just a glimpse of sterols so that you will have link and you will be able to connect the dots. So what are they exactly? They are the basic structure of sterol is a steroid nucleus which consists of 4 fused rings and 3 with carbon. Now it is a planar and rigid structure. Cholesterol, it is basically a major sterol which is present in the plasma membrane of animal cells and the plasma membrane of plant cells. They lack the cholesterol but they contain other sterols like stigmasterol and cystosterols. Now, with rare exceptions like the mycoplasma, bacterial plasma membrane, they also lack cholesterol. Lipids are basically not randomly mixed in each leaflet of a bilayer. Now, there are certain types of lipids in the plasma membrane, particularly the cholesterol and the sphingolipids, which are organized into aggregate, which are called as lipid rafts. Now, lipid rafts, what are they exactly? They are the membrane microdomains that are enriched with cholesterol and glycosphingolipids. Now, these microdomains, what they do, they contain specific proteins, and the rafton cells they appear to be heterogeneous both in terms of their protein and lipid content, and they can be localized to different regions of the cell. Now, there are two types of lipid rafts. One is the planar, and the other one is the caveole. Caveole, basically, they are flask-shaped invaginations of plasma membrane which contains the caveole proteins. And caveole is an intrinsic membrane protein, is a cholesterol binding protein. Now, lipid rafts, it has been implicated in processes as diverse as signal transduction, endocytosis, and cholesterol trafficking. Now, when the amphibatic lipids, these are mixed with water. There are 3 types of lipid aggregates which forms. And in this case of fatty acid salt, which contains only one fatty acid chain, molecules form a small and a spherical micellar structure in which the hydrophobic fatty acid chain are hidden inside the micelle. Now, a second type of lipid aggregate in water is a bilayer in which 2 lipid monolayers combine to form a 2-dimensional sheet. In third type of lipid aggregate, lipid bilayer forms a hollow sphere, which is called as a liposome. Now, liposomes, they are closed, self-sealing solvent-filled vesicles which are bound by only a single bilayer. Now, coming to the asymmetry of lipid bilayer, the phospholipids in plasma membrane, they are asymmetrically distributed across the bilayer. And the amino-containing phospholipids, they are enriched on the cytoplasmic surface of the plasma membrane, while the choline-containing and sphingolipids, they are enriched on the outer surface. Now, this asymmetry is usually not absolute except for the glycolipids. And in human erythrocyte, for example, the phospholipids such as sphingomyelin and phosphatidylcholine are found in extracytoplasmic leaflet, whereas the phosphatidylserine and phosphatidylethylamine are located on the cytoplasmic face. Now, the maintenance of the transbilayer lipid asymmetry is essential for normal membrane function. Once a lipid asymmetry has been established, it is maintained by a combination of slow transbilayer diffusion, protein-lipid interaction, and protein-mediated transport. So this is it for today. Stay tuned for more lessons to come. Thank you for watching.